I think we can all agree that our society would benefit from having better medicines, especially to treat tough diseases like cancer. However, you might be surprised to learn that we can make medicines more effective by adding a molecule that we all know and love, sugar. I'm Jessica Stark, a chemical engineer at Northwestern University, and I'm engineering bacteria to make better, sweeter medicines. The sugars that go into medicines are not quite the same as the sugar that we eat. In your body, sugars get attached to molecules called proteins. When a protein has sugars attached, it is called a glycoprotein. Glyco comes from a Greek word that means sweet, so glycoproteins are sweet proteins. Once the sugars are attached, cells in your body use glycoproteins to communicate with each other. You can imagine that cells in your body are using glycoproteins to pass notes to each other. These notes are written in the sugars that are attached to the proteins. By changing the sugars, your cells are able to send different messages to each other. It turns out that glycoproteins can also be used as medicines. In fact, they are especially good at treating tough diseases like cancer or Alzheimer's disease. However, protein drugs need to be decorated with the correct sugars, or in other words, they need to carry the correct messages in order for them to do their job fighting disease in your body. Unfortunately, we do not currently have a way to control which sugars get attached to the protein drugs, meaning that they can sometimes end up with the wrong messages attached. I am developing a technology that will allow us to control how sugars get attached to proteins. To do this, I am engineering bacterial cells to express special proteins. Let's call them builders. These builders build up sugars and attach them to other proteins. I then break the bacterial cells open, which allows me to mix the builders with other builders from other cells. Once the builders are all mixed together in the same test tube, they will band together and build glycoproteins. You can think of the builders like tradesmen or craftsmen. Each one has a particular skill, so by putting them all together in different ways, you could build different things. You could build a road or a house or even a skyscraper. By mixing the builders together in different ways, I can build different sugars. In other words, I can now write my own messages on proteins, meaning that I can now make protein medicines that carry the correct messages and have maximum disease-fighting power. As an added bonus, this technology has the potential to produce protein drugs both faster and at a lower cost than existing methods. Eventually, this technology will help us make better protein medicines both more quickly and more affordably. I hope you'll agree that that's a sweet idea.